Hello everyone. Today I would like to show you the topic very interesting. This topic mentioned about using restoring circadian rhythm in the hypothalamic paraventricle nucleus that may can reserve aging biomarkers and extend lifespan in male mice. In here they use the peso mushroom and then they check whether this mushroom can have the reverse aging and extend the lifestyle in mice model. Let's look at detail about this topic. Today I would like to show you the outline we taught today, including background, key finding, lesson learned, and discussion. Let's look at detail about the topic. In here, as you know that circadian rhythm or circadian circle in natural oscillation that present roughly 24 hour. In circadian rhythm are 24-hour internal biological clocks that regulate essential function like sleep, sleep-wake cycle, hormone release, and digestion. And these figures show how our body keep time moving from systemic regulation to molecular levels on the left hand side and the right hand side. As you can see here, on the left hand side we see that the hierarchy of the system, and S and C, that the suprachiasmatic nucleus acts as the master conductor, primarily synchronized by light, they call protic stimulus. Another one here they call non-photic signal. For example, if we do exercise or we have food, they also keep our peripheral clocks like in the liver, muscle, heart in syncs. And on the right hand side, we can see that the gears of the cell they know that transcription, translation, feedback loop. It is easy to understand, as you can see that it is symbol balance at the cone clock and B-mode 1 acts as on switch. They can buy with the e-box right here, and to dry a lot of epression. You can see that from pre-RNA, upper RNA or cry-RNA and epoch, and translation to protein and epoch the gene to the exosome. To epoch this protein to the cytoplasm. So in response to the if they if the e-box here reduce too much, like perk or cry here and perk cry here have another feedback they call negative feedback. They act on switch by to the they act on the off switch. They inhibit the clock and B-mode to reset the cycle. And finally we have another one they call drop and ref EB. They also involved in the circadian rhythm, and this one fine tune the system by regulating the B-mode 1 transcription, and together these loops ensure precise and cells sustaining 24-hour oscillation in every cell of our body. As you can see from the chart, this is the time of day at which circulating level of key endocrines, for example, cortisol, adiponectin, insulin, they call endocrine factors, peak in humans. And you know that these circle very important for homeostasis, and with cortisol are the key marker for wakefulness, where melatonin, as you can see somewhere in here at the 24 hours here for initiating sleep, and if and their disruption can impact the metabolic and physical health. Look at, you can see in here the people found that like the many rhythm like differences are different between young and old. For example, like working activity, suprachiasmatic chiasmatic nucleus are firing, lower, in older adult compared with younger one. And temperature, cortisol, melatonin, or plasma glucose are also different or reduced in older adults. And people also found that in aging population, the circadian rhythm usually dysfunction, and they can call a lot they're related to neurodegenerative diseases. As you can see from the chart, we can see that if they reduce the key gene of circadian clock like BOL1 or PERK, and I mentioned before, right, they can link with Parkinson, Alzheimer, or Huntington. And the moment thing here is the group some kind of gene related to aging or circadian rhythm, for example like SIRT1, SIRT3, or like NRF2 antioxidant gene. They also link with amyotrophic lateral sclerosis, and also a lot of diseases related to multiple sclerosis. So very important, right? But luckily, currently, people found that dietary intervention, such as like calorie restriction, including key nutrition censoring pathway involved in aging, including SIRT1, AMPK, or AMPK on NAMPT or mTOR. These pathways are tightly connected with core circadian clock components like clock's ball, clock and BOL1. You can see that as I mentioned before, right, a lot of nutrition sensing pathway here, and then they link with like metabolism and circadian regulation. Importantly, calorie restriction mathematics such as metformin. You can see here they can like involve in the MPK pathway, or ripamycin amino acid they involved in the mTOR pathway, or resveratrol you can find in rape, or NAD or amino acid they involved in the 31 here. They can mimic the anti-aging effect without reduced food intake. So additionally, many gene-related gene, I mentioned before, attention in the right-hand side here, are also expressed in the circadian manner across tissues, like in the brain, heart, liver, or like adipose tissues or the muscle. And many aging-related genes expressed in circadian manner across tissue, indicating that aging process are under circadian control. And pro-inflammation pathway like IGF-1 or mTOR here, and anti-pathway such as SIRT1 or FOXO are dynamic related over circadian cycle, highlighting the close integration between circadian rhythm and aging biology. So in this study, the new paper, right, in like from they said that enhancing circadian amplitude in mouse, they can like alleviate age-related pathology and extend lifespan. Let's look at detail about that. In here, they said that if we use a compound extract from the cordyceps mushroom, you can see that this is the mushroom, right? The structure of them, and usually previous literature also mentioned that if we use this mushroom, they have a lot of benefits for our body. For example, that they have they show that anti-hyperlipidemia, hypertension, anti-hypertension, improve renal function, anti-cancer, anti-infertility, or like antiviral, especially anti-aging. So in this study, I only focus on the anti-aging. So let's look at about the question 1. In here, they said that they want to test whether the 3AD is the time-dependent physiological effects in vivo, and whether they they see that the effects, the 3D on circadian outcome or not, right? And so in here, the first one, they perform the assay in vitro and ex vivo. And in here, in U2 OS cell, and they found that they select the PERK gene, and that the PERK gene in the core gene that oscillate every 24 hours and indicate circadian rhythm trend, and they use another they use the PERK D-look, the fusion reporter, where the luciferase track the PERK2 protein and allow real-time measurement of circadian rhythm. In here they said that like 3DA treated with 3DA have strong dependent effects, and you can see that they did a vehicle. It means that like no treatment, right? And treatment at the biological daytime, and did the CT4, and treatment with CD16 biological nighttime, right? And you see that the red and the pink here, like compare with the black one here, you can see that the amplitude is like higher, right, compared with vehicle control one. And the more important thing here, when admitted at the trouncing dose, it means that the CT4 they select this time point. They found that it significant increase the circadian amplitude around 3-fold compared with negative negative control here. And also dosing at the CT16, they did another time point here. They also found that they induce the last shift of approximately 12 hours, because here daytime, right, here nighttime, so that's why here they shift around 12 hours, along with increased amplitude around 77%. And this pattern also was found in the liver. Explain they use the ex vivo assay, and in here they show that treatment with 3DA at different time point, you can see here, and compared with vehicle control, you can see the amplitude is higher compared with vehicle control, and the effect here, like 2 different time point here, you can see that they also found they increase the relative amplitudes in the liver explain. Very important one is they said that the PERK looks rhythms in the liver explain. Usually they said that usually it's low amplitude clock was found in the mutant cell, but in here after treated with 3DA, they found that they can increase the relative amplitude. So in here they said that like 3DA robust enhance circadian amplitudes across system. Next one is they check whether the 3DA strengthen the circadian rhythm without changing thymine age mild or not. In here you can see that they did other evaluate the 3DA on circadian behavior in mice, and mice were treated via like vehicle control or 3DA around 50 milligram per kilogram IP at the light 0 hour after light on, it means that just the dark one, and even 11 hour after lights on, and every other Thursday, you can see, and then they form the well running, and you can see that what is the well running? Well running assay is the behavior non-invasive technique to use monitor voluntary physical activity, circadian rhythm, and exercise motivation in rodents. Primarily mice and rat. Animals are housed with wheel that records evolution electronically, capture data on running speed, frequency, and timing. And in the actogram, you can see that vehicle control. You can see you can see that the data discussed, right? Not so much, but after they treat with 3D, even just like 0 hour after light, or not even just dark, in the dark, and you can see that we see that a little bit like data plot in this group compared with another group. They also measure in the light dark, they light dark or content darkness, and we see that the data in this group is a little bit stronger compared with this group. And especially if you look at the after 11 hours after lights on, you can see that compared with vehicle control, we see that 3D treatment is a bit stronger and more consolidated activity rhythms compared to control. So the more important thing here is they found that quantification under both light dark and content dark conditions, so significant increase circadian amplitude, right? But the intrinsic circadian periods measured by light fraction of total activity, dark fraction of total activities, and no significant between the groups, indicate that get that 3D strengthen lock rather than shifting its timing. And the effect, you can see that the effect is more pronounced when administered at 11 hours after lights on. So highlighting that the importance of dosing time. So it means that select after 24 hours of lighting time is better to enhance the circadian rhythms. Next one is they check whether like 3D can affect the metabolic parameters in a smile or not. In here they do the same as mentioned before, right? Treat with 50 milligram per kilogram, but in here they administer at yesterday 10, compare rather than yesterday 11, right? It means that 10 hours after lights on. And in here they saw that like treatment with 3DA, like increase the energy metabolism measured by the VO2. You can see that significant difference between treatment and treated and untreated, right? Especially in the dark. And also VCO2 energy expenditures. Remarkably that the average of remarkably that we can see that for that treated with 3DA, the mushroom, and you see that they reduce the food intake and and body weight, especially the tissue weight. It means that they can control the body weight and fat mass. And not only that, the effect also found similar pattern in the glucose homeostasis measured by glucose and insulin sensitivity measured by the ACT of ITT. And they also found that they increase the behavioral stats measured by hanging time or passing time or preference for the novel object. You can see that, and also they can also check the cardiovascular function. And here they said that in young mice here, young and A treated with 3DA, and the ejection fractions and the fraction shortened. It means that ejection fraction, it means that percentage of blood per mile of the heart it beats. It means that higher, better heart function. And fractional shortness, it measures how heart muscle contract. Higher, it means that more stronger contraction. And you can see that compared with the young one, the A reduce the ejection fraction, A reduce the fractional shortness, and the but after treated with 3DA, we can see that they did recover, right? Even higher than the young one. and remarkably they increase the lifespan around 12%, measured by Kalman measure with 95% confidence interval in the graph. It means that they will extend the lifespan. This is interesting finding. So let's move on to the second question. In here they said that whether 3DA treatment can mitigate the cellular hallmark of aging or not. In here they perform a lot of assay. As you can see that the first one, they check serum inflammation factors. They measure a lot of cytokine in here, right? As you can see that, in age might increase a lot of, inflammation factor. But after treatment, we see that the overall we see that the serum infection in here less than, like the age vehicle control. Not similar with young one, but we see some, trend decline, right? Especially interleukin 6 reduced significantly, similar with young one after treated with 3DA. Not only that, we see that the DNA oxidative stress, or like ROS, or senescent biomarker, or also reduced after treated with 3DA. Remarkably, in here they measure the epigenetic aging using DNA arrays, and they found that the muscle, they reduce epigenetic age in the muscle, and even not statistically significant in lung, but we saw that some kind of trend reduced in the epigenetic age in lung. Next one is to strengthen their finding. They perform bulk RNA sequencing. In here, they do perform bulk RNA sequencing, and they say that you can see that in the A mice, without 3DA, you can see that a lot of genes so high present using the legend to see the scale, right? You can see that they high presence of the gene, but somehow treated with 3DA, they reduce the presence of the gene. Not similar with young, but compared with age, we see that they reduce, right? And and they perform enrichment analysis, and they found that these gene would link with muscle organ development, or cytoskeleton in the muscle cell. I mean, support that, like 3DA, they can improve the muscle function. Similar with muscle, they also found the same pattern in the lung. They found that, reduce the gene expression in lung compared with, vehicle control. And these gene link with a lot of, anti-survival, anti-aging pathway, and longevity regulating pathway. Next one, they check whether they will evaluate the rhythmically expressed gene in skeletal muscle and generalized across tissue. They select liver to strengthen their finding. The first one, they perform bulk analysis, and they see that around 65 up-related gene and 96 down-related gene. And in here they found that some gene here involved in the muscle contraction was reduced after a 3-day treatment, and similar with cellular calcium, but up-related gene in related to the immune related pathway was higher in treated group compared with other group. And look at the heat map, you can see that some kind of gene here was lower expressed compared with the vehicle control, and this gene would link with circadian rhythm, related to the muscle, right? And also related to the vascular mood muscle contraction, supporting that 3DA improve cardio function. They also, as I mentioned before, right? They also try to set that, like whether they can did 3DA here, they can like target not only other type cell or not. In here they select the liver, and they found that in here around 142 operated gene, 36 directed gene. And in here, similar with the muscle, they found that a lot of gene presented here reduced in treated group. Each gene involved in aging, metabolism related gene, and metabolic insulin related gene here was higher in the treated group. Similar with the one, we found that the presence of in treated group is different with vehicle control, and the gene was linked with a lot of senescence and anti-aging pathways. And the most important thing here, they also improve the cholesterol metabolism pathway. As you know that aging involved in the the group endocrine rhythm, right? As I mentioned in before, and you see that in A compared with young here, corticosteroid, testosterone, or leukotriene here would reduce compared with young one, but after treated with 3DA, this endocrine factor would higher, recover similar with young one. Next one is they check whether 3DA treatment engage in what kind of section in the brain, because that the PVN integrate and garner the multiple neuroendocrine axis. So in here they treat my with 50 milligram 3DA, and the brain would collect after 1 hour treatment. And here they select like middle O mice around 8 weeks, 8 week old mice, and they cut this brain with different section. As I mentioned before, you can see here around 8 section, and each section represent for different different section of the brain. For example, cortex, striatum, pallidum, or like some kind of like cerebellum medulla. And in here they found that among different section here, only PVJs was highly expressed the FOX positive cell. FOX positive cell did the biomarker to detect the neural activation. Like if we stand with the FOX after 1 hour, and more FOX standing, it means more neuron that recently activated. And in here they also confirmed with the ISC, and they found that with different section, right? In here vehicle control, and we see that we didn't see any FOX protein in in vehicle control. After treatment with 3DA, here we see that the presence of FOX was higher in PVN section compared with vehicle control. And they also measure the PERK. I mentioned before, right? PERK2 is the core gene of the circadian rhythm, and they extract cell from the PVN section, and they measure the PERK rhythm, and you can see that the signal in the signal of PERK rhythm in the PVN was higher after treatment compared with the control. Next one is they also check the CRY1. CRY1 also another core gene of the circadian rhythm, right? And they found that you can see that the intensity signal in young in here middle O O, and O without treatment with vehicle with without treatment, and then here treatment with 3DA, and you can see that the intensity of the O without treatment is lower compared with the middle age And in OMI with 3DA, we see that the intensity of this signal higher compared with here, but not similar with the middle age, but we can see that the signal in here is higher in compared with without treatment. And we can see that that they also measure the neuron activation again, the fog expression, and found that the fog expression was higher in the 3D group compared with treatment compared with vehicle control. Next, they also measure the calcium activity and neuronal network activity in the young mind, neuron in the young mind, and they found that treated with 3DA, they saw that the signal of the neuronal activity and neuronal network activity also higher compared with vehicle control. So let's move on to the question 5. They said that to examine whether the RUB-V2 mediate 3DE effect in the PVN neuron or not, right? In here, you know that the RUB-BV is the core gene, right? In the circadian rhythm, right? That they can modulate the E-box. So to test the necessary of the RUB-BCL2 for 3DA health benefits in A mice, they perform the PVN specific knockout RUB-BCL2 using CRISPR-Cas9 by co-injection ABV CMV SP-Cas9 with the ABV coding encoding single guide RNA targeting the RUB or control single guide negative control in the PVN. You can see that in here you can see that in the control in the control group, you can see that without treatment, you can see that the percentage of FOX here, FOX representative for the neuron activation, right here is only 1.4, 1.7, but after treatment with 3DA here, you can see that the percentage of FOX here was neuron activation here around 16 times different, right? But after the knockout the RUB2, you can see that the percentage of ROX in the vehicle or 3DA treatment here, similar, right? It means that in the PV and knockout mice, RUB-BL2 knockout mice, 3DA no longer increase the CFOX expression compared with control. So block 3DA induce CFOX activation. So it means that 3DA failed to activate CFOX in PVM of the age RUB-BL2 knockout mice. And in here, interesting finding, right? Because they check whether after we knock out the RUB-BL2, whether the 3DA can affect in other circadian or metabolic function or not. As we know that like in here single guide negative control, it means that the mice have their own gene, right? Did not knock out the RUB-BL2. We see that they increase the locomotor rhythms, and also like rhythm, especially in the light and dark condition, right? But in here, after we knock out, you can see that the data is scattered, right? And they affect the local 3D, they reduce local motor rhythms. And similar with that, we can see that they reduce the energy expenditures, VCO2, VO2, and also reduce the did not affect the food intake, and also did not affect the body weight or glucose or insulin tolerance, or especially also it did not affect the like cortisol or like inflammation measured by interleukin 6. Let's move on to the question 6. This is the final question. They said that they want to measure like they want to measure the mimic effect of 3DA treatment on the PVN using another compound they call CNO. The CNO is the CNO is not the compound. They have the same function like the 3DA, and here they use the ABV injected to the PVN neurons, and 1 month recover after ABV injection, and they will treat with saline and CNO, right? And also they will administer at the 10 day after lights on, and every other Thursday, right? And they perform well running, while handling tests, over field tests, like metabolic case. Let's look at they measure different conditions like light darks and content darks, and you can see that in the group with CNO that mimic the 3DA, they also increase the local motor rhythms in AMI. They also increase the expenditure, energy expenditure, VCO2, and also body weight. They reduce the body weight, reduce the glucose tolerance and insulin resistance. They also increase the behavior test by passing time in the center on total difference. And the moment thing here, they increase the cortisol levels, reduce inflammation and DNA methylation compared with the saline groups. So they need another model to easy understand, like the chemogenic, they call CNO here. They act similar with 3DA, and they can improve local motor rhythms, metabolic, and reduce inflammation in ASMI, similar with 3DA. So in this study, they found that 3-day treatment attenuate aging hallmarks in mind, and 3-day treatment can restore the endocrine, typically, and peripheral circadian program, and the BL2 very important, right? And they can, if we knock out this gene, the 3-DA, can mediate the anti-inflammatory benefits. So we need to keep the gene, and they found that the time activation of PVN neurons reduce the anti-aging effect of 3-day treatment. Thank you for listening.